Well, welcome back to our Bio180 lecture series. Today, we're going to continue with our study of metabolism. So over here is an outline for this whole unit. In our last lecture series, we talked about free energy and ATP and some of the things that power metabolism. Today, we're going to move on and build on our knowledge of free energy and discuss enzymes as important catalysts inside the cell for carrying out chemical reactions. Just as a preview of where we're going, we'll have one more lesson where we call what I kind of include in our biochemistry boot camp up here, learning different principles and concepts associated with biochemistry. And then we'll start moving into our biochemical reactions. So if this region is our boot camp, then we're going to start moving into chemical reactions like glycolysis, citric acid cycle electron transport. These are going to be examples of an a catabolic reaction that we'll go through to produce ATP. And then finally, we'll talk about photosynthesis in our last two lectures. And those will be examples of anabolic processes or chemical reactions that are used to build products and consume energy. But today, again, we're going to focus on enzymes and learning about enzymes, how they work, some basic principles of enzymes, how they're regulated, conditions that are important for them to work. So we're going to start off in our lecture series on free energy. We sort of introduced a diagram, and I want to come back to that, where on the y-axis we put energy, free energy, and then on our x-axis we had reactants in one column, and we had products in another column. And then we suggested that under the initial conditions of the reaction, when we looked at the free energy, the reactants might start eventually some kind of high level as we added up all the free energy of those different reactants. And we said that eventually the products themselves existed at some kind of free energy as well. And we said that the difference between those two, so the difference from my products to my reactants energy, represented the delta G of that reaction. And so we're going to return. I'm going to use this same figure. I'm going to erase the columns and just leave the bars where they started from. I'll erase that and come back to it, remembering that that difference, the distance between those two, is the free energy or the change in free energy. And when we were talking about the change in free energy, we were talking about a field of chemistry called thermodynamics. And so thermodynamics is the study of free energy changes. And today we're going to build off of this graph, and we're going to talk about a different field of chemistry called kinetics. And kinetics deals with not whether a reaction is spontaneous or non-spontaneous. That again would be thermodynamics. But kinetics deals with the rate of a reaction, how quickly it takes place. So let's do a little thought experiment to see if we might be able to understand the difference between these two. So most of you in your cupboards at home probably have a bowl full or some kind of crock or something filled with sugar, table sugar. Now, sugar itself, so if we take sugar, and we'll learn this later, and we combine it with oxygen, that can burn or combust to form CO2 and water. And if we consult the thermodynamics on this, we find that it's actually very spontaneous. A lot of energy is released when we go through this process. It's in the thousands of kilojoules per mole. So it's really quite exergonic, would be the term that we learned the other day from it. So it would produce a lot of heat. It's very exergonic. Yet, none of you are worried about running home to your apartments to see if your apartment's on fire from the combustion of the sugar in your cupboards, are you? In fact, you probably had it in some cases. Your roommates might have been having it sit on that shelf for months and months, maybe even for years if they've been there a while, right? That's because even though it's very exergonic, the rate at which that reaction proceeds is very, very slow. So while it's spontaneous, it's not very rapidly spontaneous. And that's the key. So the next question then is, if these reactions can be exergonic but occur only very slowly, what determines the rate of a reaction or how quickly it proceeds? Well, that's the study of kinetics, how quickly things go. And so to understand that, we have to come back over to our map here and understand a new term. Before, we had just thought, well, we're going to go from this energy level down to this energy level, and it was as if we drew a straight line there. But that's not actually how reactions proceed. All reactions, if you think about it, we are taking a chemical, even though it's got a high free energy here, it is at a relative stability because it exists. And so in order to convert, say, A and B as reactants into products C and D, we have to actually put energy into those because those bonds are already at some stability. A bonds and B bonds, they're already relatively stable. And so we need to add energy to them in order to destabilize those electrons in those bonds. We have to make them uncomfortable. And so we add energy to make them uncomfortable. And that takes us to a point right here that is known as the transition state of that reaction. And once we reach that transition state, the bonds are all strained and they're stressed out. They're ready for new bond formation. So then we can break them. And then we fall down and reform into our new products. And then we get a large release of energy, and that's what we call the exergonic reaction. So in this curve right here, we have this space, again from here to here, the difference in energy between reactants and products, that is our delta G. But now we have a new portion of the curve that we have to come up with a name for, and that's from the transition point down to the starting point of where the reactants A and B were. And this energy, the energy that's needed to activate that particular reaction, is then referred to as the energy of activation. Or sometimes we call it E sub A. And so when we consider the energy of activation, this is why your sugar in your cupboard at home isn't causing your apartment to catch on fire. Even though it releases a lot of energy when it converts to water and carbon dioxide, the energy of activation is quite high. And so the only place for the energy that the heat or whatever it is to come from, to feel that energy of activation, it usually comes from the ambient temperature that's coming from around the area. And it would require a very high temperature to have this reaction proceed under normal conditions. And so that's why your sugar doesn't blow up in front of you, is because of this thing called the energy of activation. You would have to excite the bonds in those sugar molecules such that they're ready to pull apart and reform into carbon dioxide and water. And that much energy just usually isn't floating around. Now, if you take a match or something or a blowtorch and add a lot of very intense heat onto that sugar, or if you heat it up on the stove for a long time, then you can get it to combust because you're meeting that energy of activation and reaching the transition state for some of those molecules. And so kinetics then is the study of this energy of activation and how to get different reactants to form and carry out their chemical reactions. Now, the next problem we want to consider now that we know about the energy of activation. So while we want your table sugar when it sits on the shelf to be relatively stable and to not sort of combust in our faces, we do eat that sugar. And when we eat that sugar, we do want to break it down. In fact, cell respiration that we talked about earlier is the whole process of taking sugar, adding it with the oxygen that we breathe in on a continual basis, and then forming CO2 and water. And that's how we're going to get our energy. We're going to take this delta G and we're going to use that to power, to fuel our bodies. How do we get the breakdown, the combustion of sugar, to work inside our bodies, which are about the same temperature as your apartments at home? There's a little bit of difference. We're a little bit warmer than your apartments are, but not much. So how do we get it to work in our bodies and not on our cupboard shelves? And that's the real challenge. And the answer to that question then is by adding what we call a catalyst. So catalysts, by definition, are things that lower the energy of activation for a particular reaction. Now, catalysts come in all sorts of shapes and sizes. The catalysts that we're really interested in when we look at the cell or the body in biology are called enzymes. So enzymes are one type of catalyst. And that's what we're going to spend the rest of our day talking about. So what an enzyme does is an enzyme will take the same reaction and it creates a curve that looks like this. So in red, we have an enzyme catalyzed reaction. And if you compare the red and black curves, then you will see that really the main difference between them is that under the effect of an enzyme, my energy of activation is much smaller. And so let's write that down, what enzymes then really do. And let's make some points about enzymes to kind of introduce them. So enzymes are going to lower the energy of activation for a chemical reaction. In fact, they're going to lower it sufficient. Most enzymes are able to lower it so that reactions occur at what we call physiological conditions. And while those vary a little bit, generally what we're talking about is 37 degrees Celsius body temperature and a pH of 7. Again, these reactions will occur at extreme conditions. So if you raise the temperature much higher than this, or if you sometimes change the pH, extreme pHs like a pH of 1, pH of 14, then you can get these reactions to go anyway without an enzyme. But to have them occur at physiological conditions, we need to lower that energy of activation so that the energy can just be met with normal body temperature. And that's the trick. And that's what enzymes do. So they're able to do that. Now, a common misconception among novice students as they're starting out is that enzymes will affect delta G. And so let's write that down. Enzymes do not affect the delta G of a reaction. In other words, for an enzyme to work, the reaction that it's going to catalyze already has to be spontaneous. Now, That's the delta G. We talked about there are ways to change delta G that we talked about in the last lecture. We can change the temperature. We can change the products and reactants, their ratio. We can couple a reaction. Those are ways to change delta G. But adding enzyme will not change the delta G. All it does is change the energy of activation. Okay? And that's a common misconception. If a reaction is not going to go forward without an enzyme, it won't go forward with an enzyme either. So enzymes do not affect the delta G. But one of the nice things about enzymes is that enzymes can be regulated. Okay? And in this sense, we're going to talk about this much later in detail, but we can turn enzymes on and off. And this gives us a way of controlling the different reactions that are going on in our body. So let's say we have about, 10,000 or so different metabolic reactions that can take place in our body. We don't want all of those taking place at the same time. So enzymes are nice because we can turn them on. The body has a way of regulating them. If we turn them on, then that reaction will proceed because the energy of activation can be met by body temperature. If we turn the enzyme off, then it returns to the normal energy of activation, and that reaction won't happen without the enzyme, at least under those conditions. So enzymes allow us to control or turn processes on or off in the body. Another thing that we want to introduce about enzymes is that enzymes are specific. Okay, so as we were saying, enzymes are specific, meaning that an enzyme itself will catalyze usually one reaction and one reaction only. So if this is an enzyme, somewhere on that enzyme will be a binding pocket or a cleft. So this represents the binding pocket. And then there will be some kind of reactant. So here's my reactant. And it is designed to fit into that binding pocket. And it's going to match both chemical charges and shape so that it kind of snuggles in there. And this reactant and only this reactant will fit into that binding pocket. It's probably fair to introduce right now, when we talk about enzyme catalyzed reactions, we actually do not use the word reactant. The reactant of an enzyme catalyzed reaction is referred to as a substrate, and that's what we'll call it from now. So enzymes are specific, so we'll add this for their substrate. And the nice thing is that enzymes also are more efficient. In producing yields of a particular reaction. So when you have an enzyme, if any of you have taken organic chemistry before, you will in the lab class carry out a bunch of different chemical reactions. And whenever you have a chemical reaction, again, to get that reaction, you have the same idea. You're trying to meet the transition state. And so what's happening when you get into your organic chemistry lab, you're going to put some reactants in maybe a beaker or a test tube. A lot of times you're going to heat that up to get the reaction to go. Again, trying to reach the energy of activation. And here's what's happening. If you could put on some molecular glasses and look inside that test tube to see what's happening, you're going to see that the heat is causing those reactants to start moving faster and faster. And the idea of the transition state is that you're getting those reactants to move fast enough that you increase the number of collisions between the reactants. And you want those reactants to come together hard enough that they kind of hit and blow their electrons, if you will, to all heck, and break or weaken and strain those bonds, and then they're going to reform into the new products. And so that's why you're heating it up, to increase the force of those reactions. But a lot of times you can get what's called competing reactions or side reactions. And so the reaction you want will happen a certain percentage of the time, let's say 40 or 50% of the time. But a lot of times you'll get these side reactions where something else happens. They hit in a wrong collision and blow the electrons up, but form into things that you don't want that are not products. And so enzymes take care of that. They're a lot more efficient. You don't get nearly the side reactions. As an example of this, there is an amino acid called glutamate. We talked about it a little bit last time. And glutamate can add ammonia to it to create the amino acid glutamine. And so what happens in this reaction, glutamate actually has the reactive center in this is a carboxyl group. So here is here's the structure of glutamate. And so what we're going to do is take a we're going to take an ammonia molecule. And we've learned about activated carbons. To make glutamine, the idea is that we're going to form this bond and break that bond, and that will form glutamine. But notice we're attacking, this was a carboxyl group, has an activated carbon right here. And we really only want the ammonia to attack this one on the left. But notice there's another potential carboxyl group right here that it could attack. And either one of these is equally probable. If I were to do this in my organic chemistry lab, I could attack this one and get glutamine that I want to, or I could attack this one and get some other side reaction product. And so let's say I get about 50% yield, which is great for an organic chemist, but still not very high. That would be like eating food and only getting half of the energy out of it, okay? It wouldn't be very efficient. When we use an enzyme, what happens is the enzyme will bind, if this is a substrate, the enzyme will bind the products and reactants, and it will cover up this carboxyl group so it can't be attacked by the ammonia, and it will position the ammonia such that it can only attack that carboxyl group. So you get increased specificity with an enzyme so that we're only affecting one reaction. And so we don't get the side reactions. So that's just another manner or way in which enzymes can be more efficient and productive for the body. So that's a little bit about our introduction of enzymes. So far, if we come back over, let's check in. We've talked about the energy of activation and then how we can use enzymes to overcome that energy of activation, and taught a few basic principles of how enzymes work, or not how they work, but a few characteristics of enzymes. Oh, I guess one more thing we should come back over here and say, probably even should have been said first. Enzymes are primarily proteins. Okay? So we learned about proteins in an earlier lecture. And so enzymes are one class of proteins. And that'll become important a little bit later on. There are, if we're being honest, there are some, about 95% of all enzymes are proteins. And then there's a small category of enzymes that are actually made out of RNA. So they're small, but some of them are really quite important. So mostly proteins, some enzymes are made out of RNA. So how are enzymes able to lower that energy of activation or lower the transition state? So let's go back and look at the classical paradigm of how enzymes work. So it's hypothesized that anytime you have an enzyme catalyzed reaction, there's actually two steps that take place. So we're going to start out with E for enzyme, and then S, that S is going to stand for substrate, the reactant. So we have enzyme and substrate, and they're banging around inside of a cell or a test tube. And the first thing that happens, there's this two-step reaction, and the first step is that we are going to form what's called an enzyme substrate complex. And so the substrate is going to come down, and remember we talked, it's going to find that binding pocket and sit down into that binding pocket. And in that, we are going to have the formation, sometimes of covalent, but usually the formation of weak bonds. And so we're going to get the formation of weak bonds. And in fact, this step we are going to refer to as binding. And then in the second step, that enzyme substrate complex is going to go the second half of the reaction. And this is where we get the conversion of substrate to product. And so this second step is called catalysis. Okay? And so we have this two-step process that takes place. And the binding is significant to the catalysis. The binding is setting the stage for catalysis. So how do we actually lower the energy of activation? Well, it's going to be somewhat through this formation of weak bonds. Remember previously, we said that when you form weak bonds, you actually release energy. We talked about this when water, you have liquid water and it forms weak bonds, more hydrogen bonds to form ice. We release energy into the environment. And so we're going to use that when we form weak bonds to make our enzyme substrate complex. We're going to release energy, and that energy is what comes up here and lowers the energy of activation. Okay? So if we're keeping track of energy, that's where we're getting some of that energy from. Now, what's happening? Why is the formation of those weak bonds so important, or how is it helping the enzyme catalyze the reaction? There are three general strategies. Not all enzymes use all three strategies. They might use one, they might use two, or a combination of them. But these are, we'll call these enzyme strategies. Okay? When an enzyme is forming those weak bonds, it's doing a couple things. For one thing, it is orienting the substrates. So let's talk about this one for a moment. Remember earlier I said if we were to do an organic chemistry reaction, we'd have our test tubes, we'd have our reactants inside, we want this reaction to happen. And so to do that, we have to get both of those reactants to come together. They not only have to hit with sufficient force, but they have to hit in the right orientation. So let's say we have to go from fist to fist, and they have to hit in this orientation. If they hit in this orientation, it won't react. If they hit in this orientation, it won't react. So there's lots of wrong ways to hit and only one right way to hit. And so that means that the reaction is going to go slowly because we'll get a lot of collisions, but a lot of them will be wrong collisions. So as we heat it up, the more we heat it up, the more movement, the more collisions, and the better the probability that we'll get the right collision. What enzymes do is they sort of, through their weak bonds, they're going to take the substrate and they're going to attract it into that binding pocket. And in that binding pocket, they're going to take it as it pulls in, they're going to orient it right into a specific formation that it needs to be. So they'll grab this one and kind of draw it in. So it's in this formation, and then this hand, they'll draw on and sit in this formation. Now I'm already in the perfect orientation, being held there by weak bonds. So now it just takes a little bit of energy to get them to hit together because they're already held in the right orientation. So that's one way that the formation of weak bonds can actually help the chemical reaction go, is by orienting the substrates in the right position. Now, the next strategy we're going to use is called catalytic acids and bases. Remember the idea in our enzyme reaction, we had to hit them together with a lot of force because what we were trying to do is get those bonds as they collide together. We're going to disrupt the electron organization in those bonds. We're going to cause those bonds to strain, the electrons to be uncomfortable. And so that's a fairly violent process. That's why we need the high energy of activation. Most of those enzymes are already somewhat stable. So how do we go about destabilizing them? We are going to, in this case, when they come down, the enzyme itself will provide, for lack of a better term, these catalytic acids and bases that are working as kind of electron magnets, so that when the substrate orients in there, the enzyme provides some magnetic pull, if you will, on those electrons that are convincing those electrons in a much milder manner to kind of pull apart or break away from that relationship that they're already in and destabilize them. So instead of the violent clash needed to kind of blow the electrons out of their current bonds, they're going to use a more subtle kind of, I don't know, psychology approach where they say, why don't you come this way and try and make things more comfortable for those electrons to move out of the way. So you can do that at a much lower energy because we're going to place those magnets right in the position next to the electrons that need to be pulled apart, not everywhere. And so that's the enzyme can introduce these catalytic acids and bases that are going to work as electron magnetics, kind of pulling those electrons out of the bond that they're in into another bond. Okay? So that's a second method. The third method we're going to use is that we are going to favor the transition state. When an enzyme, we'll often talk about enzymes binding. In fact, up here, we said that the enzyme will bind the substrate and we form an enzyme substrate complex. And while that's commonly said, it's not actually true in many cases. What the enzyme binds is not the substrate, A or B, but it will actually favor the transition state, the strained or the stressed state of that molecule. So, for example, I can't show it very well with this pen, but let's say we had an enzyme whose job was to break this pen in two. So it would position its straight bonds. We might think if this is a substrate, that you would bind and your binding pocket might look like a straight binding pocket so that it just bound the pen. In truth, that's not how the pocket of an enzyme would look. It would take those parts that would bind each side of the pen and it would tilt them such that when the pen came in, it would have to be in a strained position in order to fit into that pocket. And so in that strained position, the electrons are already being made uncomfortable. Now, why would that pen want to go into a strained position? Well, because to move into that strained position, it's going to form weak bonds and release energy. And so, remember, our weak bonds release energy. So we can actually favor the transition state. If we're sliding into the transition state, then we're just one step away from falling down to our products. So, as we finalize and review this section, we just want to remember that some different enzyme strategies that are used to lower the energy of activation is by orienting the substrates, producing catalytic acids and bases right where they're needed for that reaction, and then favoring the transition state. Now, if we come back over here, so that's some basics about enzyme function. While we were talking about enzyme function, I did remember one more thing. I want to come back over here. Under enzymes, let's put one more point down here. And that is, it has to do with lowering the energy of activation, but not affecting delta G. And that is that enzymes are reversible. An enzyme only lowers the activation of energy. It doesn't affect delta G. That's determined by equilibrium. So an enzyme will catalyze the same reaction in the forward direction and in the reverse direction. It doesn't care which way it goes. It's just going to follow or help that reaction get to equilibrium wherever that might be. In this case, we're out of equilibrium, and so the enzyme is going to catalyze it in the forward direction until we reach equilibrium. If C and D were up higher, say up here, the enzyme would still catalyze that reaction, but it would catalyze it in the reverse direction to reach equilibrium. And so I just want to point out that enzymes are reversible. Let's come over here now and talk about, I'm going to add one more thing to our list here. Enzyme, we'll call it nomenclature, naming of enzymes. So let's talk about enzyme conditions for a second. In order to carry out a chemical reaction or work properly, an enzyme has to be under the right conditions. Okay, and so a couple of important enzyme conditions that we want to speak briefly about. The first is temperature. Okay, so if you were to look at a graph of an enzyme catalyzed reaction, so we're going to put temperature on this axis. We're going to go from 0 to, I don't know, let's say 100 degrees Celsius. And then on this axis, we're going to put enzyme activity. We would see a graph that looks something like this. At 0, it would start to slowly go up. And then at some point, we would see, might not be that steep, but we'd see this pretty steep curve. And then we'd eventually hit a point where there was no more enzyme activity. So what's happening and what produces this curve? There's two sort of phases in here. In this first phase, right here, as we go upward, we're slowly increasing. And the enzyme activity is happening because we have to, remember we said binding, we have to form that enzyme substrate complex. Well, when it's cold, things aren't moving very quickly. And so the enzyme has a harder time finding the substrate. As you heat things up, the enzyme starts to be able to collide and find the substrate more easily. And so the reaction happens more quickly. Now, generally, if you were in an organic chemistry lab, this would continue. The more you heat it up, the more collisions you get, the more they find each other, and it would keep going up indefinitely. For an enzyme catalyzed reaction, though, you have some kind of optimal temperature right here because there's something else going on. So as you keep heating up, they collide more easily, they find each other more easily. But then something happens, and it decreases rapidly. And what happens on this phase is that we're getting denaturing of the enzyme. Remember, enzymes are proteins. They themselves are held together with weak bonds. And if we keep adding energy, it breaks or disrupts those weak bonds, and the enzyme will unfold or denature, and then it can't catalyze the reaction anymore. And so we have some kind of optimal temperature for that enzyme reaction, which is the highest temperature it can reach before it denatures. Now, enzymes are often designed or programmed for their optimal temperature. So for a lot of enzymes, it would go up, and then we would see it, and we would start to denature. I don't know if this is on scale, but the optimal temperature occurs right at about 37 degrees Celsius, or body temperature. And so if you go much beyond that, then you start to get the enzymes denaturing. However, not all enzymes have the same optimal temperature. For example, there is a fascinating class of organisms. If you go up to Yellowstone, that's nearby here, and you can look in those pools and you'll see these bacterial mats or microbial mats of all these different colors in the thermal hot springs up there. And those are produced by microorganisms that are able to exist at those extremely high temperatures inside those pools. Were we to go inside those pools, our proteins would start to denature. We couldn't exist there. We couldn't live. But they do, and that's because they have a higher optimal temperature. Their enzymes are held together by more weak bonds and stronger weak bonds, and so they do not denature at that temperature, and they can catalyze chemical reactions. So, different enzymes can have different optimal temperatures. So that's a temperature effect. Another condition that will affect enzymes is the pH that they're at. Again, the same kind of explanation exists for pH. If you go from 0 to 14, and you look at enzyme activity, and so this is pH on this axis, you'll see some kind of curve that looks like this, where again we have this area where we have little or no enzyme activity, and this area where we have no or little, and then we have some kind of optimum pH. And again, this is explained by this area. What happens is the extreme pHs are interfering with the ionic or the hydrogen bonding that are present in that enzyme holding it together. So too much acid or too much base will denature that enzyme and cause it to fall apart. On the other hand, there's some kind of optimum pH where the protein or the enzyme has all the right charges in all the right places, and it's able to bind its substrate effectively and carry out that enzyme condition. Now, again, as we saw with optimal temperature, although many enzymes are designed to have an optimal pH somewhere around 7, there are different enzymes. For example, the enzymes that exist in your stomach, here in green, so digestive enzymes, they're designed to work best at a pH of 2. And if you put them at a pH of 7, they really don't work very well. They will actually denature. So they're meant to form and function at an optimum pH of 2. So we've learned that you can have optimal temperatures for an enzyme, after which they start to denature, and you can have an optimal pH for an enzyme as well. And that different enzymes will have different optima. Now let's erase this and talk about a third condition that has to be present for many enzymes to work. And that is going to be cofactors. Okay? So cofactors are essential non-protein. So they're not part of, they come from somewhere else. They're not actually part of the protein. So they're essential non-protein enzyme components. Okay? Sometimes even we call them, and the name's actually not inappropriate, sometimes you'll hear them referred to as prosthetics. So a prosthetic, for humans, a prosthetic would be like an artificial leg or an artificial arm, and they carry out a function for something that the human or the body can't do by itself. And so in enzymes, you can have this prosthetic group or this cofactor that attaches to the enzyme or binds to the enzyme, and it enables the enzyme to carry out a function or purpose that it couldn't do on itself, by its own, or by itself. And so the most common function of these, it turns out that a lot of the reactions that we are going to use in biology, and many of the ones that we'll look at later in our metabolic reactions or processes, require what we call electron transfers. And those electron transfers are sometimes referred to as oxidation reduction reactions. And so we're going to spend all of our next lecture talking about electron transfers and oxidation reduction reactions. But it turns out that the amino acids that make up proteins do not do electron transfers very well. We don't have any natural amino acids that are able to take up and hold on to electrons and then give them back really well. But there are chemicals who do that really well. And so those are called cofactors. Cofactors themselves really can be split into two different categories. So one of those categories are different metals. Remember, the purpose or the function of cofactors is often electron transfer. So you need something that easily grabs electrons and something that easily gives up electrons. And so hopefully, maybe you've had enough experience to understand that metals are actually really good at doing that. That's why if you go in your home, all of the wiring in your house is made out of copper, because copper really takes electrons and it gives up electrons. So it does great with transfers of electrons. And when you transfer electrons, that's what we call electricity. So electricity is just moving electrons from one spot to another spot. And so metals are really good cofactors. If we can get a protein, an enzyme, to bind onto a metal, that becomes a complex of metals, then it will take electrons, they'll be held on that metal ion, and then they'll be given up easily. So some common metals that are used as cofactors, copper is one of them, zinc, iron, it turns out, is really good for it. Magnesium, manganese, cobalt, all of those are metals that are used as cofactors. The other category is a group called coenzymes. Coenzymes are organic in nature, and when we say that, organic in nature, it doesn't mean that they were grown without antibiotics or fertilizers and things. What it means is that they are carbon-based. And so coenzymes then are carbon-based. So they'll have some kind of carbon structure. A lot of times they'll have some kind of ring structure that has double bonds in it. And there'll be various forms or flavors of this. But this ring structure with a bunch of double bonds turns out to be able to both donate and accept electrons very well. So you'll often see some kind of structure like that present in a coenzyme. We'll learn about a couple of different coenzymes as we go into our next unit. So for example, some very common ones are going to be NADH. NADH. We're going to learn about FAD. So those will be two common coenzymes. There's one that we'll learn about called coenzyme A. It's called coenzyme A. And so we'll look at various different coenzymes. Now it turns out, well, cofactors and coenzymes may be a new word to you, new vocabulary. It turns out that they're not new concepts to you. you've actually been using or aware of coenzymes your whole life. When you were younger and your mom told you to take a vitamin, so you had the Flintstones chewable or whatever your favorite gummy vitamin was, you learned metals as minerals. So you were told to take that vitamin because it contained vitamins like B6 and B12, vitamin C. All of those are either coenzymes themselves or they get turned in the body into coenzymes. Or you took minerals like iron and zinc and magnesium in order to get you the metals that you needed to make your enzymes work. And so if you do not, to show how necessary these cofactors are, having the enzyme, if it requires a cofactor, you have to have that cofactor, you have to have that vitamin. And throughout history, particularly during the Middle Ages and those times, when they didn't have as balanced a diet, you would often find diseases associated with the deficiency of different enzymes. So for example, scurvy is a disease that causes bleeding gums and some other factors. Sailors used to get it a lot, and it comes from a lack of vitamin C. So you have to take vitamin C to get your metabolic processes to work. If you don't, then you end up with these deficiencies. And so coenzymes are essential components. You have to have it or you don't work. Another one that's essential, iron. So if you've ever heard of the term anemia, like sickle cell anemia, or there's different types of anemias, all of them refer to a low blood count. And a lot of times that can be due to a lack of iron in the diet. So lack of iron in the diet can produce anemia. You don't get the red blood cells that you need, which require iron in order to bind oxygen. So vitamins and minerals, cofactors, that's another condition. The availability of cofactors is another condition for enzymes to work. Now, let's move on to our next topic. I'm going to skip ahead a little bit before moving to our final topic of regulation. And let's talk about enzyme naming or nomenclature. Enzyme names. Most enzymes that you see are going to be binomial, which simply means that they have two names. So, like humans, they're going to have a first name and a last name. So the first name, well, let's do the last name first. The last name is usually a description of that enzyme activity or what it does, its occupation. We have human last names like this. For example, if any of you have the last name of Smith, that probably derives somewhere in your lineage from an ancestor who was a blacksmith or a coppersmith, maybe a tinsmith, but some kind of smith. So the last name Smith implies an occupation. If you have the last name of Tanner, then Tanner was someone that would tan hides and do work in leather. So Tanner is an occupational last name. Cooper, if you have a last name of Cooper, Cooper was a barrel maker. And so you have different occupational last names. And in enzymes, we also have occupational last names. And some of those, just like in humans, we have some very common last names like Smith or Cooper or whatever a common last name might be. We have some common last names in enzymes too, and these are ones that we'll see over and over again and that we'll come to be familiar with. One common last name is kinase. Kinase is like the Smith of the enzyme world. So kinase is an enzyme that's going to hydrolyze or use ATP somehow, and it does ATP transfers. And we'll see what those mean as we go on in a later unit. But kinases do ATP transfers. We have a class of enzymes called hydrogenases, dehydrogenases. As the name implies, hydrogenase. These remove hydrogens from things, but we're going to see that these are involved with NADH and NAD plus electron transfers, or really any kind of electron transfer. Another common one that we will see is phosphatase. Removes phosphates. We'll see if you guys can guess this one. We have dealt earlier with a whole section called isomers. So there's an enzyme class or a family called isomerases. As you might guess, these convert one isomer into another type of isomer. So convert isomers. So those are 4 of them. Notice that all of these are ending in the same suffix, ASE. And so if you see the ASE suffix, then you know that you're talking about some kind of enzyme. So this is a suffix that's reserved for enzymes. Not all enzymes use it, but many do. I guess we'll do one more. There's hydrolase or lyase. Those are enzymes that cleave. We'll learn about synthetase or synthase, ATP synthase. Synthase taken from the root word synthesis, so that's something that synthesizes ATP. The first name of an enzyme then, so those are all last names. The first name then identifies a substrate or a product of that reaction. Of the reaction that it catalyzes. So, for example, we could have multiple dehydrogenases. So we might have an enzyme. So one enzyme might be malate dehydrogenase, where malate in this case happens to be the substrate of that particular dehydrogenation reaction. Or you could have succinate dehydrogenase. So those would be 2 different enzymes that share the same last name. So they're doing the same or a similar chemical reaction, but they're acting on different substrates. So the first name will usually identify the substrate, or in some cases the product, because remember they're reversible. And the last name then will be a description of the enzyme's activity. So that's a little bit about naming enzymes. So we've got enzyme nomenclature. Now our final topic, let's talk a little bit about enzyme regulation. Well, there's some various strategies for enzyme regulation. There's actually a couple different ways of regulating enzymes. So 2 of the most common that are used, well, maybe we'll mention a couple of them. So one way that enzymes can be regulated, we'll call this one temporal regulation. Temporal regulation is what the cell will do. It will either build the enzyme, or when it's no longer needed, it will destroy the enzyme. So when we need the enzyme, we make it, and when we don't need it, we destroy it. And that's referred to as temporal. It's temporal regulation, okay? And so that's not terribly efficient method. That means you've got to build it up and then destroy it. So usually this is for long-term regulation techniques. There's another one called spatial regulation. This one is kind of a theoretical form of regulation. All this means is that you have, it's confined, the enzyme is confined to a compartment. So as an example of this, you have, we learned earlier about the lysosome, that this was a digestive center inside the cell. And so you have digestive enzymes that are regulated by their location in the lysosome. We don't find them other places, so that's how the cell regulates it. It takes those enzymes. They're on all the time, but they're on only in this certain space all the time. So that's an example of spatial regulation. So while these happen, they're not going to, I don't want you to worry about them too much. By and large, as we go about our further discussions of the cell, we're going to talk about two primary forms of regulation called phosphorylation and allosteric regulation. And then we're going to talk about a third one that isn't used as much actually in the body, but we'll talk about competitive inhibition as a form of regulation. So we'll go over these three. Let's talk about the first one, phosphorylation. Really, both of these have the same basis, and that is that enzymes have an active and inactive conformation. And that's the word that we use for it, conformation. Conformation is just a big word that means shape. And so an enzyme, I'm just going to draw it up as an enzyme will have an active shape, like a square, and that it can convert to an inactive shape, like a circle. And if we draw this a little bit more specifically, we might say in the active conformation, remember we said it had a binding pocket, and that binding pocket would fit the substrate, right? So in the active conformation, that binding pocket is open. So now we have open pocket. And here in the inactive, the pocket probably still exists, but it will have changed its shape. So now we try and take our substrate, and we find that that substrate can't go into, it won't fit in that binding pocket anymore. And so what will happen is enzymes will be able to fluctuate between the active and inactive states. So they just have this ability to change between these two shapes, kind of like a Dr. Jekyll or Mr. Hyde, or I guess now we'd say Bruce Banner and the Hulk. And so the enzyme can fluctuate between these two different states, just like Bruce Banner can activate between these two different states. But that's not regulated, so it can go between these two different states, but we want to be able to control it, right? And this is what Bruce Banner was always searching for, is control over being able to go between these two different states. And so what we're going to do is need to have something that could freeze it in one state or the other. And in a phosphorylation regulation, what we'll see is that one of the states, adding a phosphate, and this is almost always added from ATP, will freeze, quote unquote, the enzyme in one state. So then enzymes can be added by taking a phosphate. So I'll take ATP and I'll form ADP. And then the phosphate will get stuck here on the enzyme and it will freeze it in the active form. And so as long as I'm phosphorylated, I'm active. And then if I ever remove that phosphate, then I'll naturally fall down into the inactive state. So if I'm phosphorylated, I'm active. And if I'm unphosphorylated, it will kind of fall down energetically into the inactive state. And so that's a form of phosphorylation or regulation by phosphorylation. Now, there are some enzymes